So in this video, I'm going to talk about transcription. And so the overall process, it takes the DNA and essentially turns it into mRNA as a final result. And so it's pretty much a way to transfer the information from DNA to the outside of the nucleus. And so let's talk about that a little bit. And so here's a good diagram. We won't focus on down here. We won't worry about that. That'll be another video. And so the promoter right here is the region of the DNA in which the RNA polymerase binds. So promoter, RNA polymerase binds to that site. Actually, let me use a different diagram. This one's pretty complicated, so let me clear this. And so I just pretty much just drew it. So here's a promoter. And then the next section of the DNA right here is a regulatory sequence. And what that does is it's the site of binding, so binding, regulatory proteins bind there, and binding of regulatory proteins. And so what the regular proteins do is they influence the rate of transcription. So how fast or how slow it goes. And it's also found in a variety of locations. And so here, in the blue, is the transcribed region. And what that is it contains the information that specifies an amino acid sequence. So it's essentially the whole, sequence of DNA you're trying to copy for the certain protein. And then finally you have here in the green the terminator, which signals the end. It signals that the mRNA will be cut there. And so in total, 10% of your DNA codes for RNA molecules. And those are also called non-structural genes. The rest code for proteins, which are called structural genes. And now let's get into how transcription occurs. So first what happens is the gene unzips. So first gene / DNA unzips, and the base pairs are exposed. And so this serves as a template for the mRNA. So essentially, the mRNA forms based off of one strand of DNA. And remember, when you're making RNA, uracil replaces thymine. And so eventually, what you're going to result with is a complementary mRNA molecule to the DNA strand. And Rna polymerase is the molecule that synthesizes the mRNA. And like DNA polymerase, it only works in the 5' to 3' direction. And since only one side of the double helix is being used, that strand is called the coding strand. So the coding strand is strand that's being used. To make mRNA. And so eventually, once you finish transcription, that mRNA actually cannot be used to make proteins yet. It has to be modified. And so let's talk about that. So right after mRNA is made, it's called the primary mRNA. And that's before any modifications have been done to it. And so it's modified before it leaves the nucleus, and it is spliced. So we'll talk about RNA splicing. So some segments will be cut out, and so those are called introns. So these are segments that do not specify amino acids. So as a result, they're going to be taken out. So they're spliced out by the spliceosome. The spliceosome complex in the nucleoplasm, which is the cytoplasm of the nucleus. So anyway, introns don't specify for amino acids, they're cut out. Exons are the important ones. They specify amino acids, AA, and they're the ones that are kept. And so what happens is the spliceosomes cut out the introns and reattach exons to produce a smooth coding sequence. And this, once again, occurs in the nucleus. And so some other modifications have to occur to the final mRNA. And so what happens is there's something called a methyl guanine cap, and this is added to the 5' end of mRNA, the finished mRNA with only exons, to help it bind to ribosomes. And then a polyadenine tail is added to the 3' end of mRNA in order to help it move through the nuclear pores. So through the nuclear pores, which essentially means it helps it get out of the nucleus and into the cytoplasm. And the final result is a mature mRNA transcript. And that whole process is called translation, turning DNA into a usable form that the cell can use to make proteins.